Yo, what's going on, guys? Welcome back to our GCSE psychology journey, and today we're going to be looking at the neuron structure and the function. So let's get it. So starting off with, let's look at different types of neurons. So what is a neuron? So this, as we can see on screen, is kind of the things that make up a neuron. So we've got the nucleus of the neuron, we've got the cell body or soma, we've got dendrites, and these reach out to the other neurons to communicate. We've got the myelin sheath, which acts as an insulator, and that helps the action potentials or the electric impulses travel between neurons. And we've got the axon terminals. The direction that the message or the action potential travels is from left to right, as we can see here. Every type of neuron has these elements to it, but they're all arranged differently depending on the type of neuron. And we're going to have a brief explanation of how some of those may look when we look at different types of neurons. Now, roughly in the adult, there is about 86 billion neurons in the human brain, just so you guys know. So we've got sensory neurons, and the function of sensory neurons is to carry messages from the sense organs to the central nervous system, or the CNS. That's the brain and the spinal cord, if you guys remember our previous video. They're also called afferent neurons. Key features of these are that they have long dendrons, they have short axons, and the cell body is located along the axon in a ganglion. So you can see that they're slightly different from the example that we just looked at. We have relay neurons. The function of relay neurons is to pass messages between the sensory and motor neurons within the central nervous system. These are also called interneurons. An example of this is in a reflex arc. So the relay neuron connects the sensory neuron to the motor neuron in the spinal cord. Key features is that they have short dendrites and also short axons. And interestingly, these are only found in the brain and spinal cord. We have motor neurons. The function of motor neurons is to carry messages from the central nervous system to the muscles or glands to cause a response. These are also called efferent neurons. An example is when we pull our hand away from heat, the motor neurons send signals to our muscles. Key features is that they have short dendrites, long axons, and the cell body is at one end. So here we can see an image of our pre and post-synaptic neurons. So on the left here we have our presynaptic neuron, and within our neurons, when it comes to the process of synaptic transmission, we have things going on. So I'm going to explain those as we go. So on the left here we've got our presynaptic neuron, which is number one. And number two is our postsynaptic neuron. So the presynaptic neuron is the neuron that sends the signal across, and the postsynaptic neuron is the neuron that receives that message or action potential. And what is that action potential? Well, the action potential is when neurotransmitters are sent across, and we've previously looked at different types of neurotransmitters, things like serotonin, dopamine, acetylcholine. That process of one sending to the other is called synaptic transmission. The neurotransmitters themselves within the presynaptic neuron are held within these little vesicles, and each vesicle has a certain amount of neurotransmitters contained within it. Once that action potential is fired, then those neurotransmitters that we can see here as the little red dots pass across the synapse or the synaptic cleft into the receptor cells, which we can see here as number 5, the receptor cells on the postsynaptic neuron. And that space in between is called the synapse or the synaptic cleft. And that process is synaptic transmission, when the action potential fires off and allows those neurotransmitters to go from our presynaptic neuron to our postsynaptic neuron. We have excitatory neurotransmitters. Now we've got excitatory and inhibitory neurotransmitters. We're looking at excitatory neurotransmitters. Now these neurotransmitters increase the likelihood that the postsynaptic neuron will fire an action potential. So when we just looked at that synaptic transmission process, an excitatory neurotransmitter increases the likelihood that that action potential will fire. They make the inside of the postsynaptic neuron more positive or depolarized, moving it closer to the threshold for firing. An example of this is glutamate, and this is the most common excitatory neurotransmitter in the brain. Inhibitory neurotransmitters. Now, excitatory increase the likelihood that the postsynaptic neuron will fire an action potential, whereas inhibitory neurotransmitters decrease the likelihood that the postsynaptic neuron will fire an action potential. They make the inside of the postsynaptic neuron more negative or hyperpolarized, moving it further from the threshold. An example of this is GABA, or gamma-aminobutyric acid, and that's the main inhibitory neurotransmitter which is found in the brain. So glutamate is the main excitatory neurotransmitter, and GABA is the main inhibitory neurotransmitter which is found in the brain. So now let's have a look at Hebb's theory. So Hebb's theory was proposed by Donald Hebb in 1949, and it explains how learning occurs in the brain through strengthening the connections between neurons. The core idea behind this is that cells that fire together wire together. Hebb suggested that when two neurons are activated at the same time, the connection between them becomes stronger. Repeated activation makes the connection more efficient, and over time, this creates a neural pathway that represents learning. He called this process synaptic plasticity. Some of the strengths behind this method is that it's supported by biological evidence. It explains learning at a neural level, is influential in neuroscience and also in cognitive psychology. Some of the limitations: it can be viewed as slightly oversimplified. Learning involves more complex brain processes than just firing and connection, and neuroplasticity is based largely on theory rather than direct human experimentation. And it does not explain all types of learning, so learning through things like insight. Structure and localization of function in the brain. So now we're going to look a little deeper into the different areas of the brain and neuroanatomy. So the brain is divided up into 4 lobes. So here we can see we've got blue, yellow, green, and red. We also have in white there we have the cerebellum, but we're not going to be looking at that as a lobe right now. So this first blue area, this is our frontal lobe, and the frontal lobe is responsible for things such as our thinking, speaking, memory, and movement. Now within the frontal lobe, that is divided up into other sections, but we're keeping it very simple at this stage right now, because we have things like our prefrontal cortex, which is our most developed part of our brain. Down here we have our temporal lobe, heavily responsible for things such as our hearing. We have our occipital lobe in red at the back here, and that is heavily responsible for vision and color. And up the top we have our parietal lobe, which is heavily involved with things such as touch, language, taste, and smell, so our senses. Now this is a very oversimplified explanation of what these areas do. We do have, of course, association areas, as well as primary functioning areas of the brain. But these are the 4 major lobes which are divided up by major sulci within the brain. So our frontal lobe, our parietal lobe, our occipital and temporal lobe. So our motor area of our brain is located within the frontal lobe, and it controls voluntary movement of the body. Example, things such as walking, writing and speaking. The left side controls the right side of the body and vice versa. So our left hemisphere controls the right side of our body, and the right hemisphere of our brain controls the left side of our body. The somatosensory area is located in the parietal lobe, and it processes information from the senses such as touch, pressure, pain, and temperature. It also has a sensory map of the body, our sensory homunculus. Our visual area towards the rear of the brain it's located in the occipital lobe. It's responsible for processing visual information from the eyes, such as color, shape, and movement. Our auditory area, which is located in our temporal lobe, and it processes sounds, including speech, music, and other noises. Cognition and language. So here we have highlighted in white Broca's area, and this is responsible for our speech production. Damage to this area can lead to what is called Broca's aphasia, and that leads to difficulties in forming speech. We also have Wernicke's area, again highlighted here in white, and this is highly responsible for our language comprehension. Damage to this area can lead to Wernicke's aphasia, so we can have fluent speech, but it can be quite nonsensical. We're going to look at Wilder Penfield's study of the interpretive cortex, and this is Wilder Penfield, who we can see on screen now. The aim of his study was to investigate the function of the interpretive cortex, which is part of the temporal lobe, by electrically stimulating the brains of conscious patients during surgery. His method? Well, it was conducted in the 1950s at the Montreal Neurological Institute. Patients undergoing surgery for epilepsy were kept conscious, so they only had local anesthetic rather than general anesthetic. Penfield applied mild electric stimulation to areas of the temporal lobe. Patients were asked to report their experiences. So what are the findings? When the interpretive cortex was stimulated, patients reported vivid, detailed memories, for example, hearing music, voices, conversations, and feeling like they were reliving past experiences. Some experienced hallucinations, distortions of time. However, Not all patients experienced memories. Memories were sometimes fragmented or dreamlike rather than accurate recordings. So what did Penfield conclude from this? Well, he concluded that the interpretive cortex plays a key role in memory storage and retrieval, and that memories may be stored permanently in the brain, and that electric stimulation can trigger experiential responses. What are some of the strengths of this study? Well, it had high ecological validity. These are real medical procedures. Direct evidence linking specific brain areas to behavior. It was influential in understanding localization of function. What were some of the weaknesses? Well, it was a small, very unusual sample. So these were epileptic patients, so it had low levels of generalizability. It was an artificial simulation, so it may not reflect normal memory processes also. And some experiences may have been reconstructions and not true memories. Alright guys, hopefully that gives you guys a better understanding of the different types of neurons and that important study from Penfield and also from Hebb. If you haven't already, which I hope you have, make sure that you guys like, make sure that you guys subscribe. We'll continue our journey on our next video, so I'll catch you guys in the next one. Peace.